This is the first part of your genetic engineering notes. So let's go ahead and start. So genetic engineering is the deliberate tweaking with and editing genes, and they can be in people, like with cancer research and treatment, or they can be with other animals or plants or fungi or bacteria. But it's the deliberate making of changes in the genome or the sequence of DNA in any organism. So one way to do this is, well, it's not really genetic engineering, but one way to change the genome is selective breeding. And this is where we pick and choose traits or desirable traits and features that we want in, say, dogs, and we breed them so that their puppies or their offspring have those traits or are more likely to have those traits as well. This is not gene editing. This is not genetic engineering because we're not using any kind of technology here. Rather, we're just telling and essentially forcing individuals to mate with each other. But it is nevertheless a way to change the genome. So we'll talk more about artificial selection in our evolution unit. But this is how we got dogs. Dogs are not a natural quote unquote animal. They were artificially selected for by us, by people. Same thing with a lot of domestic cats and plants. A lot of the plants that you eat for dinner, perhaps, are not wild natural plants. They are artificially selected for by humans. So that's an example of selective breeding. Hybridization is essentially when you're crossing two dissimilar looking individuals and you try to have offspring that have kind of the best of both traits. So there's a little bit of trial and error here. Again, we'll talk more about this as it's necessary in the evolution unit. Inbreeding is when you are crossing similar organisms. This is how you get, for example, I'll talk about dogs again, how you get purebreds like German Shepherds or like a purebred horse or thoroughbred. And basically everything that we're doing here on this slide is based off of what the individual looks like. We don't really know at this level what their genes are, but we do know what their traits are. And we're like, okay, well, I like your trait and I like your trait, so you guys are going to mate. We'll talk about the genomic consequences of that later, but this kind of selective breeding is really only done by most people based off some kind of physical, apparent, or obvious characteristic. Although I guess you could, if you want, actually sequence the genome and then do selective breeding, but usually it's not. All right, so actual genetic engineering is going in and changing the DNA. So this is really the main point of the slides. This is where you would go in, physically cut and remove pieces of DNA and replace them with a working copy or other gene. So organisms that have had this done to them are called transgenic, right? Trans meaning across and genic meaning gene, so genes from a different species. So here's a glowing tobacco plant. From memory, I think this is GFP, green fluorescent protein from a firefly or lightning bug. It's either that or a jellyfish gene. I have to go back and check, but that's what that is. In this case, a gene was inserted into the genome, and there's no harm done to the plant. You can actually do this with animals as well. You can Google transgenic glowing rabbit, and you can find videos on glowing bunnies. And the bunnies are perfectly healthy. They just make this protein that glows, that gives off light. So some more vocab here. Clones, genetically identical cells or organisms. They have exactly the same DNA. So if you have an identical twin, you are clones of each other. You're not a clone in the sense that you were made in a lab. So I guess I would say the word clone is for like if you like in a lab setting made another individual exactly the same. So actually, I'd say identical twins, yes, you're identical twins, but I wouldn't call you clones in that regard. I'll show you some examples of clones, like Dolly the sheep was our first actual clone, where we went in and deliberately copied the DNA. Well, we didn't copy it, but we manipulated it such that it gave a different organism gave birth to a clone, and we named that clone Dolly. And I'll show you that in a different video. So the tools of genetic engineering are, again, it's kind of the backbone of the stuff for this set of notes. So the first thing you have to do is extract DNA. We didn't do this lab, and I might post a video showing you how it's done. It's actually super, super simple. So assuming that you have extracted and isolated the DNA, the second step is that you have to cut it. We'll talk about how that works, but you're going to use what are called restriction enzymes. Actually, this technique is a little bit old, but it's still kind of part of the main content for the course. These are our scissors. Think of restriction enzymes like being biological scissors that specialize in cutting DNA and RNA. Then you're going to separate out the pieces that you've cut, and that's called electrophoresis. And then you can make copies of anything that you've cut to mass produce it, kind of like you would make copies at a copy machine. That's called polymerase chain reaction. So let's walk through these steps. So when you cut the DNA, you're cutting them using enzymes. Remember, enzymes are proteins that catalyze a reaction, right? They reduce the activation energy and they facilitate this reaction. So this reaction is actually breaking the backbone of DNA and the hydrogen bonds that run down the middle between the bases. So these enzymes come from bacteria, right? So we isolate them from bacteria, so their names are going to sound like bacteria, and I'll give you an example. But the point is that they cut at very, very specific places. They don't just cut willy-nilly. They cut at a specific point in the DNA. So let me give you an example. So here's a restriction enzyme, EcoR1. So here's where I told you the name is going to sound like bacteria. That's E. coli. So they got EcoR1 from E. coli. This means E. coli restriction enzyme number 1. So this was the first restriction enzyme they got from E. coli and named it EcoR1. But EcoR1 cuts the DNA. So here's a DNA sequence up here. Should be an arrow. Specifically at this site. So wherever it sees CTTAAG and its complementary strand here, it will cut. And it'll cut the backbone between the G and the A at the end. That's the covalent bond here that it cuts. It cuts all of the hydrogen bonds between, in this case, the A's and the T's, and then cuts the backbone on the other side, so that covalent bond, on the opposite strand. So that's going to cut the DNA in half. And if you notice, the sequence is a palindrome. It's the same forward on one side as it is backward on the other in this case. I guess it's not a true palindrome in the sense of the word, but if you go CTTAAG from left to right on the top strand, you'll go CTTAAG from right to left on this bottom strand. So palindrome is the wrong word. They're opposites of each other, and they're always going to be that way. So what you get is, because you've kind of cut across the DNA molecule, you don't get a nice clean cut up and down. You get a cut that leaves this little fragment here, this single-stranded fragment. This is called a sticky end. By the way, these restriction enzymes cut everywhere they see the sequence. So if they see the sequence again, like over here on the right, they'll do the exact same cut. So that's why you have 2 cuts here in this example. So these sticky ends are important because when we glue the DNA back together, that's where it's going to happen. All right, oh, I'll go back here really quick. And notice that you have three fragments. You have this fragment over here on the left. You have this big fragment here in the middle, fragment number 2, and then you have this third fragment over here. So if you make 3 cuts on a piece of DNA, you'll get 1, 2, 3 sections. So we can take those sections. It's just like if you cut a piece of paper. If you cut the paper twice all the way across, you'll have 3 pieces of paper. So you can separate those pieces of paper or pieces of DNA out, and that's what we do next. So this is called gel electrophoresis. So go ahead and read this over on your own. You can pause the video, and then I'm going to show you on the next slide what this looks like. All right, so I'm assuming that you've paused and read the steps. I think it's easier, though, to kind of just look at the picture and talk about the steps in context. So this gel is a thick, viscous gel. Yes, it's mostly made of water, but it's kind of like the cubes that we used to show you diffusion. Remember in that video where the cubes were pink and they were kind of like rubbery almost? It was kind of hard to see it in the video, but if we were in person, you would feel it there. It's like a gel. It's like a ballistics gel. It's the same kind of thing. So it's a thick gel-like substance. And it's hard for DNA to move through it. But if you are a small piece of DNA, you can move through it quicker. So small, you go through quicker, and you make it further through the gel. And that's the key point here. You actually separate by size. The smaller pieces of DNA will travel through the gel further because they're small enough to fit through the gel. The bigger fragments are kind of too thick to make it through the gel, so they don't travel as far. But here's our gel. And what you do is you put the DNA at one end of the gel. These little holes here are called wells. They're exactly that. They're little holes, and that's where you pipe the DNA into, which is what they're doing in this picture here. And then when you put a power source on the gel with the negative end where the DNA starts and the positive end where it will finish, and then turn this power source on, because DNA is negatively charged, when you turn this power source on, the negative charges are going to repel each other, and that's going to push DNA towards the positive end of the gel. That's pretty cool, right? And again, the smaller pieces of DNA will travel out further, and the shorter ones will kind of stay closer to the negative side. And after, I don't know, after half an hour, 45 minutes, if you turn the power source off and you take the gel out of the little holder, and you stain it so you can see the DNA, you'll see little bands. The bands that are on the negative side are the larger bands, and the bands that are on the far side, the positive side, are the smaller bands. And this reads just like a barcode. If you have something you buy at the store, if you scan the barcode, it's unique to that item. So if this were my DNA in this gel, this code, this barcode, this striped pattern would be unique to me. And we can actually use this barcode to identify people if we know their DNA. All right, so the last thing we can do here is, again, since we've now separated the copies of DNA out, we can take those pieces of DNA and make millions and millions of copies of them, because if you are a forensic analyst, perhaps, and you're solving a crime and you have sampled some DNA from the crime scene, you might not have enough to give out to a bunch of different laboratories. So you might have to make copies of it so that each lab has their own copy, so that they can test it for different characteristics. So this is why you would want to make copies in the first place. So this is called PCR, polymerase chain reaction. You might notice that the enzyme we're using here is called polymerase. And it's gonna behave like a chain reaction. It's gonna get bigger and bigger as we go on. So let's take a look at this together actually. So the first thing that happens is you have basically a hot water bath with nucleotides in it. A's, T's, G's, and C's. They're free floating just around in that water bath. So we think of it as a soup. And when you put the DNA into that water bath, the DNA that you want copied, the high temperature of the water and the solution will break the hydrogen bonds between the two bases. So your double-stranded DNA, if that's my double-stranded DNA molecule, will separate into two single-stranded molecules. And that's just because, again, high heat breaks the hydrogen bonds between the bases. All right. Then we let this solution cool, and we add DNA polymerase into it. And what that does is DNA polymerase makes a complementary strand on each half of the original DNA molecule. So we started with one DNA molecule, and then we end up with two. And I apologize for this line coming across. I don't know why it does that. So now we have two strands of DNA, two molecules of DNA, which is 4 strands, whereas to start with, we had half that. So if you do this over and over and over and over again, you heat and cool, heat and cool, heat and cool, you can go from 1 to 2 to 4 to 8 to 16 to 32 and so on molecules of DNA. So you get an exponentially increasing amount of DNA pretty fast. Right, so here's a visual of it. You start with your double-stranded DNA. High temperature separates the two strands. Each strand has a complementary strand built onto it. And then you just repeat that over and over and over and over again. So you very, very quickly get potentially thousands of strands of DNA after just a couple rounds, right? So we did talk about, doing this in the context of forensics. That's one of the main ways you can do this. But if you noticed, we are heating a solution that has an enzyme in it. Right? Polymerase is an enzyme, and just like hydrogen bonds, enzymes are very sensitive to high temperatures. They denature. Think all the way back to Unit 2 when we talked about enzymes and how pH can denature them, so can temperature, right? So why does this type of enzyme, DNA polymerase, work in the hot water baths that we use in polymerase chain reaction? Why doesn't DNA polymerase, in this case, denature? Well, it doesn't denature, the answer is right here, because it comes from bacteria that evolved in hot springs. So again, the line, I apologize for that line. If you have evolved in hot springs, the normal quote unquote temperature for you, the tolerable range for your enzymes is what we would consider to be hot. So we use those enzymes from those bacteria that evolved and lived in those hot springs so that their DNA polymerase doesn't denature. And it doesn't matter that it's bacterial DNA, it doesn't matter that it's a bacterial enzyme. It doesn't matter because DNA and the code, the genetic code, is universal. The DNA polymerase that works in your body works in the bodies of bacteria, right? So that's why we can use it. So that brings us to the end of the techniques behind a lot of genetic engineering. I will leave you with a picture of what a PCR machine looks like. That's kind of really all it is. If you take AP Biology and we're back in person for it, then you'll actually be able to do gel electrophoresis and make your own bands. So all things to look forward to, and I will see you in class, and I hope this helped.